Hi, my name is Larry Grossman. I'm the president of the Maryland chapter of the American Chestnut Foundation. I'm here at the Baltimore County Agriculture Center in their greenhouse. We're in the process of planting 2,000 chestnut seeds in our quest to develop a blight-resistant hybrid. As you may know, the American chestnut was wiped out by a blight accidentally imported from China about 100 years ago. We are working to develop a blight-resistant hybrid through a process of back-crossing between American and Chinese parents. It takes us on the order of 6 generations of crosses to achieve that. Each generation takes about 10 years, 8 to 10 years, to grow up to maturity to produce enough chestnuts for us to use in our program. So once that's happened, then we are out in the field harvesting chestnuts and then planting 2,000 of them here in this greenhouse to allow us to grow them up, screen them for blight resistance in the summer, and then plant them in our orchards in the fall. So our Maryland chapter has about 200 members. Nationally, the American Chestnut Foundation has about 5,000 members. We're nonprofit organizations. We don't pay salaries. We do this on a volunteer basis. And we're hoping in a few years to see if what we've been doing is successful. So if you want to join the fun, give me a call or send me an email. Google Maryland Chestnut Foundation, and my email address is there. Contact me that way.
My name is Bruce, and I'm with the Maryland chapter of the Chestnut Foundation. And what we're doing here is planting hybrid chestnut seeds to determine how resistant they are to the chestnut blight. And by June, we expect them to be about this high, and we're going to do a micro inoculation with the blight fungus and watch them over the summer. And those that survive through the whole summer without wilting will be the most resistant. So that not just tells us which would be the most resistant trees and we can plant them in the field, but it also tells us which families have the highest survival rates in the family. So we can compare the different mother trees to each other. So we're doing 2 things. One is we're selecting the trees for the next generation of breeding, and the other is we're selecting the progeny that are the strongest in those families.
So I see we have little white tags in all of these. How many different varieties or how many different strains are we planting here today?
Okay, so these the tags represent the mothers that they come from, and there's probably 22 to 24 of them. Some of them were tested last year, and some haven't been tested until this year.
And this is a fairly new iteration of this breeding program, is that right, the way we're doing it today?
What we did was we planted the seeds directly in the ground, and we'd wait 5-6 years until they were large enough to inoculate. But that requires taking care of the trees for all that time and maintaining the orchard, and then we would cut down 99% of them. So what we're hoping to do is get the worst 80 or 90% out of the population before we put them in the ground. That'll give us a smaller population of better trees that we have to tend.
And we hope to have the hope is that we'll have a chestnut that can be produced and survive against the blight in how long? The hope, anyway.
Yeah, that's a good question. So it's probably a few years off yet. These trees are already more resistant on average than an American because on average they are about 13% Chinese. And they carry some of the resistance genes from Chinese chestnut.
Yeah.
It would probably take another generation or 2 of breeding to get really highly resistant trees, but that may speed up if the genetic work that other parts of ACF are doing tells us what the specific genes are, and then we can use genetic tests to inform our breeding. But right now, we have to just do it randomly.
Yeah, and from the meetings and some of the papers I've seen, the genetics, that have worldwide, not just specifically for trees or anything, have accelerated this work dramatically.
Right. So we can do things now that we couldn't afford to do even 5 years ago, and it's done very routinely. And it's already starting to tell us, for example, the theory was that the 2 or 3 genes were involved in the resistance of the Chinese chestnut, and now we're seeing that there are far more parts of the chestnut genome that are involved. And that makes the breeding more complicated, but it also helps us hunt down what those genes are.